pick up this first because this is something probably everybody in the world probably now recognizes, right? This is the what we call the glamour molecule in my world, actually. It is so iconic. People have it on their T-shirts and all sorts of things like that. But the thing you hear less about, but is actually the hard-working part of the cell, does most of the work in the cell. Do you know what it is? It's a molecule that's called a protein, right? So proteins are very humble, you know. They do all the work, and they most of the time are not very glamorous. If you look at what I see in my hand, you probably won't see this on T-shirts any day now, right? It's really the idea that this is so recognizable, and this really isn't. This video demonstrates a teacher technique called a hook. A hook is a question or a demonstration that makes students curious and interested in learning more. Most importantly, a hook does not require a student to have prior scientific knowledge to participate in the classroom activity. The purpose here is to level the playing field so all students can participate. That's because so many teachers ask questions that require terminology, giving students from rich backgrounds an advantage. So the hook broadens participation and interest in the whole class. Specifically, this hook with proteins asks students to make visual observations about the model. In this way, students can learn several important characteristics of real proteins by their observations. It is easier to work through the observations in a particular order by asking certain questions or prompts. I will give you the 4 prompts that you might use to get you started in the class. I hope you will try this in your class. Let's do this. Let's have the model tell us something about proteins, okay? So I'm going to, this is participatory. I'd like for you to pretend that you are just going to look at this. You're not going to remember all the big vocabulary you know about proteins. So we'll just see if the model does a good job showing what proteins are like, okay? So right now, just watch for a few seconds. I'm going to fold this up. I'm going to turn it around. And I'm going to ask you, just by looking at it, what is the overall form of a protein? How would you describe a protein if you just had to describe something about the overall form? What would you say? Yeah?
Well, if you want, like this is linear.
Great. I heard that it's linear. That's great. It is a molecule that's kind of long. That's a great part. What else did you notice? Yeah?
Like the individual parts stick together.
Great. So you've noticed that it has different individual parts, and you're noticing they stick together even when I'm doing this with it. So how else? That's great. What else would you add?
It's like flexible, movable.
Excuse me? I just didn't hear you.
It's like flexible.
Yes. Okay. So we've got this really nice flexibility, and it's going to respond to its environment as to how it folds up. So that's number 1. That's terrific. We now know that proteins are flexible, long molecules that can fold up into different shapes. In fact, you've probably heard about that lock and key thing that enzymes do, you know. They need to have a little pocket so you can make these shapes. Right, good. Okay, now let's see what else the model can show us. Look real closely. I'm going to just pull apart a couple of the subunits, all right, here. Okay, what do you notice now about the subunits? What do they have in common? What do these subunits have in common? Somebody else want to try? Just what you see, that's all. What do you notice that these have in common? Yep.
They both have a label.
They both have a label. Good. All right. So in fact, when we're working with them, we want to learn their names a little bit. So that's useful. But let's think about something else. Yeah?
The shape is the same.
What is the shape? What parts are the same that you notice? Maybe color? The color parts which parts are the same? This one and this one? Okay. So both of them have a black part and a gray part. That's great because actually when they go together, you can see that all along the chain we have the same repeating parts. And I love it because actually this part is that amino acids say their name. They're really cool. The black part is going to be the amino group, and the gray part is going to be the acid group. So amino acids always say their name, and they're really easy, and they're the subunits of the protein. All right, so we've got subunits named amino acids. They're flexible long chains. Can you just tell me something else you notice about the the chains right now? There's something really obvious, right? What do you think?
Someone repeating?
Yes, you're getting the black and the gray parts that repeat. What about the part in the middle? What do you notice about those? Yep.
They're different colors, different shapes.
They're different colors and different shapes. So the colors, this is really important, the colors represent the chemistry. So all of the yellow ones are going to be hydrophobic, all right? When you're building with this, you're going to need to know that the yellow ones are hydrophobic. So what does hydrophobic mean? Yeah?
They're scared of water.
They're scared of water, okay? So when we try to fold these up and there's water around, the yellow ones are going to try to hide more in the middle, okay? Away from the water. The colors also, how many colors do we have? I told you about the yellow one. How many colors total in this group, in this whole thing? Can you see? Yeah? 4 different colors, so we have 4 different kinds of amino acids, okay? We've got hydrophobic ones, which are yellow, and the other colors are all hydrophilic, all right? And also what we noticed from this is that you mentioned they're different shapes. And the shapes are actually representing the atoms. So if it's a really long chain here, it will be a long one, all right? And if it's a very short chain, it'll be a very short side chain on the molecule. All right, so just by looking at the molecule as a model, we picked out a couple of main things. Want to look up here? All right, proteins are flexible chains that fold up into different shapes to do work. They're amino acids as subunits, and there are 4 different kinds, and they each have their own individual shapes with the labels on them, which was really great to bring out. We're going to bring out the sets for you. We're going to make it so that you will share a set. If you're a team at one table, you'll share a set together, okay? And Amanda and I are going to quickly bring these around, and you can actually start by opening up the booklet, okay? And you want to clear your decks because we're going to be doing a lot more building, okay? Here is one essential tool that teachers need to know in order to manage the classroom. So this is the kit care record, and you can see, you can get it downloaded online and designate whether it's going to go in the DNA, protein, or tRNA set, and the kit number. So students should receive a kit that looks like this with the folded kit check inside of it. And at the beginning of class, student teams should be instructed to fill it out, looking at each of the pieces and recording if anything is missing. So the date, their names. If there's nothing missing, they should make sure to write the word OK in here so we will know it's been checked. So they filled out the form, and they do it this way by checking the label here and making sure the right pieces are here. And working with a partner, this can take just 1 or 2 minutes. So it will get to be a habit at the beginning of each class. And then put the pieces back together at the very end and put it in. The end of the class is usually pretty rushed, so we don't end up doing a kit check at the end. But you can see when the next class comes in and they write their names on it and they check it out, they will know, there will be a record whether there's anything missing after it's used. So the purpose of this, again, is that you will always be able to have a good class without interruptions for any missing articles in the lesson plans. So the same system can be used for the DNA and RNA kit. So you can see it here. You put the number on and mark it that it belongs with the DNA and RNA. And in this case, you have the label on the inside. And sometimes these smaller pieces we put in a bag so you can take them out if you're not using them. They're only used in some of the advanced lessons. Okay, so then the last thing I'd like to point out is the tRNA sets that can be used by the teachers. If you are sharing your set, sometimes it's really nice to have a teacher record of who's used it and how they've been set up last. So anyway, this is something that you should not ever be without, because this really helps you manage things easily. Start by opening up your booklet like this. Just the booklet so far, okay, please? Stick with me. We're going to stay together, okay? We're going to work together. So this, I'm holding the booklet up. When we use the booklet, we don't want you to fold it back like this, because it turns out most of the information you need is kind of open on 2 pages like this. So if you'll open it up that way, that would be great. I just want to clue you into how to use the booklet. If you open up your box right now, it's kind of like a candy box. I think of it this way. Have you ever bought those kind of chocolate candies where they have all different kinds, but you know that one of them you really hate, like you really hate the jellies or whatever? Okay. So what we want to do is help you find the right amino acid when you need it. So if you open up the box, you can actually see in the lid in the back, it shows you where everything is, like this, see? So basically, if you want to look for glycine, you can look right up here and see that glycine is, can you find glycine on your own set? I know it's yellow, it's GLY. And I look down here and I can actually find it in the box. So why doesn't everybody just pull out, let's go for a MET, an M-E-T, all right? See if you can find it and pull it out. That'd be great. Each person pull out a MET. That'd be really great, okay? We're going to explore a little bit about how the amino acids work around here, okay? Can you find a MET, M-E-T? And each person hold one. That's great. And in the book you can actually see, I like each person to hold one. So if you haven't got one in your hand, please hold one in your hand. Great. I'm really hoping you'll start to fool around with it, because one of the things you'll notice, like someone over here is noticing, that the side chain is very flexible. In fact, it does twirl around. And we had to do that. I actually designed the sets, and so one of the things I had to do was try to make it so the amino acids behaved the way they really do in nature. So if you have a MET there, use good. You can actually see that when you hold it up, it can actually do little cartwheels. Okay. So this is natural that the molecule is more flexible in this spot. Okay. So that's what we want to show. So would everybody hold it up? I'm just checking for knowledge here. Hold it up by the amino end. Raise it up. Black end. Hold it up by the amino end. This way. Got it? I'm just checking. Okay. Got the amino end? All right. Hold it by the acid end. Hold it by the side chain. Okay, good. And your biology classes, did you call it an R? An R group? Okay. So that's the same thing, all right? So the side chain or the R group. Good. So we can see that MET is kind of a long chain, and it's quite flexible. Okay, we're just going to get a little bit more familiar, because I'm going to have you build certain things just pretty soon. So what I'd like for you to do is to find the smallest yellow and just hold it up. Each person find the smallest yellow. Just to get more familiar, please. This will help you in the long run when you're looking for it, okay? Put the box in between the two of you so you can both reach in. That might help. Yeah, that'd be good. Okay, good. That does it spin too? I'm just checking. Yeah, it actually spins. That was such a shrimpy one. If you look up here at the chemistry, you can actually see that gly only has one hydrogen. Its side chain is teeny tiny, right? And it's teeny tiny in the model. Okay, right now, would you find the largest yellow? And this one, you have to look at the chemistry. So how did you decide between phenylalanine and trip? P. Which one is the biggest? Did you find the chemistry here? Because these are pretty close to being the same. But then I had to make them the same shape because they're pretty close.
I think it's this one.
But it's the trip, right?
It's because it has more hydrogen, right?
Yeah. It has two rings like this. So these all have carbons on the end, so they're really bulky. So big side chains are big and bulky. They kind of have that big cross thing, and we're trying to make it so that when you fold it up, it will be like the real amino acid. It will be either small and easy to fold into something, or actually more difficult. I have the best challenge. Can you find one that does not spin? There is one amino acid that is really different from all the others, and it does not spin.
Yeah.
Did you find it?
Connected to the amino.
Amino end, you know about that? That's great. That's good. Yeah. Yes, you found it, proline. Did you know about it before?
No, I just noticed that.
Okay. Excellent. That's good. You looking at the chemistry? Did you find
because it's
proline?
connecting the two things.
Yes. Yeah, that's good. So would you put those back in where they belong? This is the next challenge, to get them back, okay? You work together as a team. It'll be really quick. That'll be great. Nice work. Okay. Just to find out now, we're looking now at the hydrophilic ones. These are all the ones that are going to love to be near water. Instead of sunbathing, they're going to be water bathing for sure. They're going to like water. So what's the smallest green you can find? Just look up at the chemistry yourself or in the book. SER? Good. What's the largest pink? I think it's glutamic acid, right? Glue? You got that? Okay. All right. And the only blue is basic and pink is acid, just so you have that in your mind now. Acid and base are pink and blue like you'd expect, like acid and base in chemistry, right? Okay, good. So I tried to make the colors suggestive of what they are. So the basic ones are blue, the red ones are, pink ones are. All right, good. Okay, it's now time for a little bit of great designing, okay? Each person, so sometimes I say each person, sometimes I say each team. So each person is going to make their own designer protein 4 amino acids long, so you get to hook them together. And I'd like for you not to choose this one. I would like to just see what you feel like building, okay? So just do this as a little easy starter for building proteins. 4 amino acids long, each person builds their own. You can do whatever you want for color, size, shape. Be creative. Each person's building their own, okay? Go for it. Nice. So you remember what bond you're making when you click them together? Like that? What's the name of the bond? Do you may know?
So you want to build one as well? Yeah. It's a team of 4. Any 4 you want.
That's right. You just made peptide bonds. Don't make them any longer than 4 and don't connect the ends. Just make it a little chain, okay? Proteins always stay a little chain. If you look at both ends, you'll notice they're different. There is an amino end and there is an acid end when you hook them all together, right? So I'd like for you to hold it up by the amino end right now. Everybody just hold it up in the air so it's hanging down like that, like that. And the reason why we're doing this is we are deciding, you know, that chemists have to decide which amino acid is going to be number 1 when they want to give an order, right? We want to say, I want you to build this protein. What's the order? So you have to decide which one's number 1. The amino end, this is real important, the amino end is actually the one that's going to be number 1. So you're all holding up by your number 1 amino acid. And now if you hold it up, we're going to check around the room to see if anyone built the same protein as you. Okay? So hold it up by the amino end, and we're going to look around the room. And hold it so it's dangling so you can see what the number 1 is. And look around the room. Can you check out, see if anybody built it like yours? If you see the colors close, check the labels. I want to find out if anybody had the same inspiration as somebody else. Remember to hold it up by the black end, which is the amino, and then you can tell which is number 1, right? Are you sure nobody has one like you? So we've determined that we just let everybody make whatever they wanted, and nobody made the same thing. And I'd like to show you, the reason why I had you do that was because I wanted to show you that proteins are really cool. DNA has lots of variations possible, but proteins do too. There are about 20 different kinds of amino acids. And if you put them together, we get to do a little math right now, okay? Are you ready? Let's see. I have 4 amino acids here, okay, like you do. And I could choose any one of 20 to put in here, okay, in the top one. And then I could choose any one of 20 to put here. So the variation I have is 20 times 20. How many ways could I make one just too long? 400, right? And then I could go with 20 more possibilities here. And how much would that be? 20 times 400? 8,000. And another time, 20 times more. Look at this, 160,000 different ways of making a little protein, only 4 long. And most proteins are, how long do you think? Just guessing. 100? Really, they're big. Most proteins are pretty big. We made a little small one. So if they're hundreds long, can you imagine what the varieties are? It's really amazing. So I have a lot more respect for proteins nowadays since I actually went and did a little math and got an idea. So right now what we're going to do is we've learned a lot of just basics and we haven't had a chance to actually get into seeing some bigger proteins. So I'm going to show a little video right now, but I'd love for you first of all to put your own 4 back where they belong because you'll need to find them again, okay? And we've really got a lot more context now for working with proteins.
The MIT Edgerton Center presents Introduction to Proteins, to be used with the MIT Edgerton Center protein set. What is a protein? Every protein is a folded chain. Each link in the chain is an amino acid. There are 20 different amino acids. How are amino acids different? All amino acids have 3 parts: the amino group, the acid group, and the side chain. Each amino acid has a different side chain. Some side chains are large. Other side chains are small. Most side chains can spin, except proline cannot spin because proline's side chain has an extra bond. Why are the side chains different colors? A side chain's color represents its chemical properties. Yellow side chains are hydrophobic. Hydrophobic means water-fearing. Pink, blue, and green side chains are hydrophilic. Hydrophilic means water-loving. Green side chains are polar or partially charged. Pink side chains are acidic or negatively charged. Blue side chains are basic or positively charged. How do amino acids form proteins? Amino acids bond together. An acid group bonds to an amino group to form a peptide bond between amino acids. The order of amino acids is very important. A different order makes a different protein. The number of possible proteins is huge.
I oftentimes look at school books and I see that the cell looks like it's got nothing in it. There's nothing there. You see a single cell there, but it's really completely full of lots of different molecules, which include things like proteins. And they look like blobs, but as we know now, they're really folded chains. And the 2 chains both have, and the chains all have different ends. So we're going to build this protein as a team coming up next. You and your team will build this one. And we're going to do some folding of the chain. And before we do, I'd like to give you another short video about how to fold, which will be useful to you, okay?
The MIT Edgerton Center presents Protein Folding in Water, to be used with the MIT Edgerton Center protein set. Every protein is a folded chain. A protein's job requires a unique shape. Protein folding begins with the correct sequence of amino acids. Build the protein chain on page 11. Keep the black amino end to the left as you build. the I of each amino group should be showing. Otherwise, the chain cannot bend. Turn all the amino ends so the I's show. Turning the ends helps the model to fold. Check the rest of the chain. How will the protein fold in water? Hydrophobic side chains hide from water. Hydrophilic side chains are attracted to water. Gently fold the chain using this information. Hydrophilic side chains turn to face the water. Hydrophobic side chains turn towards each other on the inside of the protein. The hydrophobic amino acids are clustered together in the center. The hydrophilic amino acids are on the outside of the protein. This protein chain is folded using only one rule. Several rules are required for the unique shape of each protein.
Why don't you begin right now to do the building? It's on page 11. And you need to put it in this order. Make sure that you keep the amino end to the left as you build. The black ends to the left, okay?
So remember, when you build, you want to make sure you're keeping the amino end to the left. Yes. So you just have to turn them all around.
All right, everybody has a chain and it's looks good. We've got our amino ends to the left. Now we have to, and have you checked the eyes? Remember we were going to check the eyes? Right. I'm always tempted to want to put the two ends together, but proteins don't do that, so we've got to act like they do. So that's sort of out of their what they like to do. So just check right now that the little eyes are showing, because what we're going to do is pretend that there are water molecules all around, okay? So if there are water molecules all around this protein, the yellow ones are what kind? Remember what the word is? Hydrophobic? Excellent, okay. So they will want to be more in the interior of the protein. And the hydrophilic ones, oh, they love the water and their side chains are going to flip out towards the water. So there are lots of correct answers. We just like to see that you show what you know. Would you and your partner choose to fold it up as if there's water around? And Amanda and I will walk around and talk with you about it, see how you're working it out, okay? Just a quick teacher reminder that one answer for folding proteins in water is shown on page 12. However, there are many possible answers, and some of those were shown in the video. So please make sure that you choose this opportunity to move around the room, visiting all the student teams and giving them guidance. This should be an opportunity as a teacher to engage with students more individually. One of the benefits of the models is that students can show what they know and also what they don't know. So a teacher can very easily assess the situation and give advice.
Hydrophobic, right, which means they don't like water, right? So when you don't like something or you're scared of it, what do you generally do? You stay away from it, yeah. So if this protein is surrounded by water, where do you think these yellow water-fearing ones are going to want to go? Yeah, exactly. So in the interior, as far away from the water as they can get.
We have the yellow ones crowded together here. And if these would defy gravity, they would be out in the water like that. That's great. We've got them isolated and out like that. That's good. Proteins have more than just this one rule, this one about hydrophobic, hydrophilic. We're just trying it out right now. And if they were following all the rules, everybody would have the same shape at the very end. So you start off with this order and it would end up being the same shape. We know we've got lots of proteins in our cells, but right now, how does the protein get there? So we'd like to think a little bit more. Most people, when they think of proteins, they think of food, right? I mean, I used to think of proteins as food pretty much. And so can you name some proteins, can you name some foods that are high in proteins? I'd love to hear from some people I haven't heard from before too, so yep. You know some proteins, right? Foods that are high in proteins? Yep. Do you have a food? Oh, he does, all right. What'd you say? Meat for sure. That's one that comes out very easily, yep. Okay, almonds, sure. Nuts are good. Yep, I'm glad somebody said a fruit or a vegetable kind of plant-based thing as well. I'm really glad about that. Yep. Beans, yes. Particularly good also. Yep.
Eggs.
Eggs, yes, for sure. Eggs.
Bread.
Bread. Bread has protein, but some people wouldn't call it high in proteins, but it's, yes, you could always say bread. Mm-hmm. What'd you say? Broccoli, not so high in proteins, but okay. It's all right to try it out. Yeah, I wouldn't call that high. Yep.
Just like generally dairy foods.
Some dairy foods, yes. When you think about it, when you were a baby, where did you get all your proteins from? Was milk, right? So it had to grow, yeah. And anything else coming to mind? We're doing great. Yeah. Fish, thank you. Fish are up there too. Yep, they are too, actually. So we have all of those possibilities, but I'd like to show you just one quick way in which we think about how proteins get made into parts of you. So in a little summary, I'm going to pretend here that I'm going to eat eggs, all right? You ready? Okay, here we go. So I've got eggs here and egg protein, and I'm going to have it, I had it for breakfast, okay? Are you ready? Okay, so now it's in my stomach, and basically it gets broken down by into its individual amino acids. So I've got these small amino acids, and they're small enough so that they can get taken into the blood everywhere, right? So they get taken, let's say, into the bloodstream, and they get taken out to the muscles over here. And when they're here, the DNA here in the muscles give the order of the amino acids. That's the job of the DNA. It's a storage device. It basically gives the order of the amino acids. And so what we get is a different order here. And when it's a different order, it's a different protein. Okay? So in other words, I don't make egg proteins here. That would be really bad, right? What happens is you're making muscle cells. Muscle cells need to have muscle protein, and the DNA in that cell puts these together in a different order, and that's how you get your own muscle cells being made with more proteins, okay? All right, and I know that the cell, the body does make some of its own proteins, but it really likes to keep the energy down. These are convenient, they're really available, so your body puts them together in different order, and that's how you make different proteins. There are two options for teaching how cells make new protein. You can choose to do the demo I just did with eating an egg protein and then seeing what happens to the amino acids, the change in order that makes for muscle proteins. But you can also do an entire classroom activity that is found in the booklet. It is on page 13 through 18. And this is very interesting because the students will make an egg protein example, then take it apart and rearrange it into a protein that is found in hair called keratin. Teachers tell me that this is a very good example and a very engaging one. What we're going to do right now is I'd love for you to take apart your proteins that we made as a test. We're going to actually start to build a protein that has a job to do, okay? And we have to be really precise and fold it in different ways. We're doing great. We've got a lot of good start on proteins overall. Now we're going to build one that has a job to do, a particular function. We're going to model a cell membrane. Do you remember when you learned about cell membranes? Was it in middle school? Around then too, yeah. So it was one of the earliest places people learned about, paid attention to proteins, was actually thinking of the fact that they could make the cell membrane work better because you have to get things in and out of the cell. So right now on your table, this is like the top of a cell. The table is like the top of a cell. And you can actually see that there's a little membrane. I'm folding the sides up so it looks like it's got, what are these things here? Do you remember what this is called? It's the real reach back. What is it called? This is a cell membrane. What are the molecules called here? Yeah? Phospholipids, right. So we've got the phospholipids here, bilayer here, and we're going to show that the proteins are going to go all the way through the bilayer, okay? And we need to build some specific ones in the right shape. You can actually see that the protein in this diagram, here's the phospholipid bilayer. You can actually see that the protein that we're going to do has about 10 different turns. And we're only going to make a short little protein, okay? And because later on I'm going to give you a gene and you're going to build the protein again from that gene. So it's going to be a little bit shorter, but it's going to pass through all this hydrophobic area in the middle here.
The MIT Edgerton Center presents Introduction to Channel Proteins, to be used with the MIT Edgerton Center protein set. Channel proteins create pores in the cell's membrane. Pores control how molecules move in and out of the cell. This channel protein has 4 chains, 2 alpha chains and 2 beta chains. Together, they create the pore. The pore allows molecules to enter the cell. Each protein chain is folded into a helix. The chains are surrounded by the cell membrane, and the cell membrane has 2 different sections: a hydrophobic middle and hydrophilic surfaces. Your cell membrane mat folds up to create a 3D model with a hydrophobic middle and hydrophilic surfaces. The cell membrane's function depends on channel proteins.
The alpha chain is on page 20 and 21. So you and your partner, one will build the alpha chain and one will build the beta chain, okay? And then if you look at your table, you actually have 4 little chains that can come together. And you can see on this that there's 2 alpha chains and 2 beta chains. So you're going to insert them into the cell membrane the way you think they go, okay? So that's the objective at this point.
The MIT Edgerton Center presents How to Fold a Helix, to be used with the MIT Edgerton Center protein set. Step 1. Build the protein chain. Make sure the amino end is to the left on the first amino acid. Step 2. Turn the amino groups so the eyes are showing. This is important for being able to fold the protein. Step 3. Fold the met upwards. Step 4. Fold the chain into a J shape. Step 5. Fold the chain over to make a box. Step 6. Lift and stack the last 2 amino acids on top of the box. Check your helix. All the side chains should be pointing out. Here are some things not to do. Don't connect the ends of the protein chain. This does not happen in nature. Don't break the protein chain. The folded protein must be complete. Let's watch the correct way one more time. Step 1. Build the protein chain. Make sure the amino end is to the left on the first amino acid. Step 2. Turn the amino groups so the eyes are showing. Step 3. Fold the met upwards. Step 4. Fold the chain into a J shape. Step 5. Fold the chain over to make a box. Step 6. Lift and stack the last 2 amino acids on top of the box.
Why don't you start off by building the chains first, and I'll give you a little bit more explanation about how to make the helix in just a moment. Okay?
Here, you do the.
So which one's going to make the alpha and which one's going to make the beta? Okay, good. Remember, what's the word to remember about which end is number 1? It has to have amino in there. Good, okay.
Oh, I think you were right the first time. Yeah, there you go. And then fold kind of the rest of the end up. Yep, like that. And then you're going to fold the top over. Great. All right, now this is the hard part. You're actually going to pick these 2 up, yep, and place them on top. Perfect. Yep. That's perfect. So now you're just going to do the same thing with the beta chain.
Yeah.
Good.
All right, let's do it.
Yeah.
Second one.
So same thing. Starts with
Met and then
pro,
and then sir.
then glue. Then out. It's in.
Questions?
So if you make like the helix longer, is it just a repetition of the same amino acids?
Actually, it's a great question. Is it a repetition of the same amino acids? I think after the end of the exercise, you'll probably be able to tell me what you think it is, okay? So hold that thought and I'll come back to it again. Many students will be confused about what to do first and how to orient the helices into the cell membrane. And this is okay. I just would like to point out that it may take a few minutes for students to think more carefully about what is surrounding the proteins in this case. So please provide them with hints, move around the room, and live with a little bit of ambiguity so that the students can learn and teach themselves to think more about things and make the right choices. Good. Can you pick it up? The trick is picking it up too, I know. You want to put your little side chains back out. See, like this.
Does it matter which order, like which way it's facing?
I'm getting asked the question, which way is it facing? Maybe you want to think about that. I'd love for you to yeah, use your own judgment, how you think it should go. There you go. Nice. You have to get yours over there too. Can you do that? Maybe the membrane will scoot in your direction. Transport. Good. What you thinking? Explain what you're thinking.
Something about the
She's
yellow.
thinking about which way to put it in.
The yellow is probably, I don't even know. Probably shouldn't be on the outside. It probably should be on the inside because it's hydrophobic. Hydro.
Well, let's think about it. I think you're bringing up a good point. Where is the hydrophobic area? What's all around here? The liquid.
That's
Is it water?
oh, this is hydrophobic.
This right.
Yeah. Yeah.
Right?
Right. Yeah. Good. Good. You're keying a lot of people to start thinking about where the hydrophobic areas are. So all these turns are near the hydrophobic area. All the turns are near the hydrophobic area. So which way do you think they would be?
The yellow facing outwards.
Maybe the yellow one's facing outwards. You want to try it? Try to just make a little spin. Okay. And see where the positions of these turn out to be.
How do you think bees would want to orient themselves to be happiest if you got all hydrophobic molecules out here?
Oh, so the yellow would be turning towards the outside?
Yep, go ahead and turn them. Perfect. I'll turn this one. There we go. And that one's turned a little bit. Perfect. So now we have all the hydrophobics contacting the hydrophobic part of the membrane, which makes it happy. And we have all the hydrophilics actually creating this pore in the center, which is where things like water or ions or other charged or partially charged things can go in and out. And in addition, blues are positive, pinks are negative. So these are actually attracting each other as well. So this is how the structure of the protein actually works.
The protein right now is in the cell membrane, right? And the cell membrane it's fitting into a lot of other molecules. And what are those other molecules like? They are hydrophobic or hydrophilic? All those hydrophobic tails, right? So the yellows are not going to want to be on the inside. I fooled you if you thought they always went to the inside, because we have to really see about what's surrounding the protein, right? So there's no water surrounding these proteins. They are deeply embedded into the hydrophobic layer of the membrane. So would you spin your proteins to make them align so that the hydrophobic yellow parts are pointing out toward the outside? This is incorrect on the front here. If you were just following up there, you would discover that the result actually should look more like this. Want to look at this one? Want to look up for a moment? You see this one? The yellow ones are hydrophobic sides are on the outside because they're in the hydrophobic membrane. See it here? And what's really cool is if this goes into this position, what happens is, what are the amino acids on the center going to be? Are they hydrophilic or hydrophobic?
Hydrophilic.
Yeah. They're hydrophilic, and ions have charges, and they can pass more really easily through the cell membrane itself, okay? And in order for this to work, if I may pick up somebody else's right here, let's see what the order of the amino acids has to be in order for this to work correctly. You look up here, everybody. Do you notice that there's a pattern? What's the pattern that you see in the order here? You may see a pattern? Yep. Hydrophobic, I'm just repeating what you said. Hydrophilic and then hydrophobic, right? And you have to have that pattern in order to have one whole side be hydrophilic and the other side to be hydrophobic. If you're looking at yours right now, you can see that one side is always going to end up being hydrophobic and the other side is going to be hydrophilic. And therefore, the protein will need to fit into the membrane so that the hydrophobic sides are toward the outside. And we had an unfinished question up here, so perhaps we can all think about it for a moment. The question was whether the protein sequence exactly stayed the same as it went up. If it was longer, what would you expect the next amino acid to be, right? What kind? So if we hold it up again, we had 2 hydrophobic, 2 hydrophilic, 2 hydrophobic. What would you expect the next one to be?
Hydrophilic.
Right, and then 2 of them and keep going. So the question about whether it had to be exactly the same, how would you answer that?
No.
No, but?
But it still repeats in a pattern.
It still makes the same overall pattern to be successful. So if there was a mutation when you stop and think about it and you got the wrong thing here, what would you expect to happen to this channel protein?
Wouldn't work.
Maybe it wouldn't work so well, okay? So that's why the DNA is so important not to have any errors or mutations in it, because it could affect the function of this channel protein. And in fact, the channel protein is something that we'll come back to later on when we actually make some genes, and you'll build the exact same protein actually starting from genes and go all the way through.